Hi, thanks for joining our Synergy webinar series, where we designed the next hour to discuss complementary techniques for vaccine development life cycle. My name is Julie Chenuin. I am your facilitator, and I'll be monitoring any questions and comments that you have in the background. We will have a Q&A session in the end. Okay, so let me introduce you, my very, very favorite speaker, Dr. Kevin Dahl, whom I respect on a professional and personal level. So Kevin Dahl is the founder of Particlese, a consultancy dedicated to assist pharma clients with particle and spectroscopic challenges. Kevin received his PhD in physical chemistry with a focus in electronic spectroscopy from the Penn State University. He continued his education with an NRC postdoc appointment to the U.S. Naval Research Lab in Washington, D.C., where he worked with vibrational spectroscopy. He then joined a major particle sizing company as a tech specialist, where he worked with light scattering microscopy and business development with both large and small molecule pharmaceuticals. After that, he joined a biopharma CDMO, contract development and manufacturing organization, to stand up a specialized particle lab that provided contract and analytical services to the larger biotech companies. He established Particlese in early 2022 and to continue his work in the field. So next, I think I need to let Kevin introduce our other amazing speaker, Dr. Linda Kidder, as they've known and worked with each other for many, many years.
Thanks, Julie. That's very high praise. I really appreciate it, and I'll have more of the same for my friend Linda Kidder. So when I started out of my postdoc, Linda was actually the person that hired me into my first commercial job. Linda is extremely well known in the spectroscopic community. She's been working at a very high level there for a number of years. She started off with a PhD in chemistry from Johns Hopkins University and followed that up immediately with a postdoc at NIH under Neil Lewis, who's another well-known figure in the spectroscopic community. After that NIH appointment, Neil and Linda both founded Spectral Dimensions, which is where I met them. Spectral Dimensions was then acquired by Malvern Panalytical, and that's where at that point, Linda's commercial career really took off, where she had various jobs at places like Shimadzu, Brightspec, and then finally with HORIBA, where she is now their life sciences business development manager. Linda has been involved in quite a bit in the community, including General Chair of SciEx Conference in 2020, Governing Board of the Society for Applied Spectroscopy, or SAS, and then was also a past board member of the Covalent Society. She's one of the most genuine and wonderful people I've ever had the opportunity to work with, and I know she's got some very exciting things to share with you during her presentation about A-TEEM. Now, as Julie mentioned during my introduction, so my background is really in spectroscopy and then particles. And I'm going to be talking today more specifically about the particle side of vaccines. But before I get to that, so as Julie mentioned earlier this year, I founded Particlese LLC, and I bring to the table about 20 years of particle and spectroscopic experience in pharma. I specialize in consulting really across the entire breadth. I've done the R&D side of things. I've done the business development side of things. I've worked in a GMP capacity. So technology consulting, data document method consulting, these different facets of the business are something that I specialize in. I also have access to some experimental consulting and services, or experimental consulting services with one of the universities here in Colorado. And then for at least selected instruments, I'm available for doing training on those systems. And if you'd like to get in touch with me, I'd love to have a conversation. You can reach out to me at kd@particlesellc.com. So to begin with, I love to start off with a quotation. It's a bit hackneyed, but certainly I think looking at people who've worked in this field and some of the things that they have said in the past can give us a good foundation for going forward and understanding some of what's being done now. And Jonas Salk, I don't know that there's anybody bigger I think even most laymen know exactly who he is, or at least associate him with vaccination from his work on polio. And what he said was the reward for work well done is the opportunity to do more. And I think that's especially relevant nowadays because of, oh, what, about 3 years ago, we had a global pandemic. And within 12 months of the start of that global pandemic, we had multiple vaccine products that have been developed and brought to at least EUA approval for use in the broader community. And that really sets the stage, I think, for kind of a renaissance in in vaccine production, and probably heightens the expectation for people in the broader community as well for what can be done with these pharmaceutical products. Now, what are the challenges in vaccine development? I'm sure the audience here knows as well as I do. So obviously discovery, formulation, which I include delivery and formulation here, storage and transport, which is really more around freeze thaw, but with some of the cold chain issues that are experienced in developing countries, this is really a critical piece of getting vaccinations out to people. Stability, so this is more long-term stability. In-use stability, so from the point of preparation until it goes into a patient, and then release testing. So these challenges, I think, with the exception of discovery, there are very clear places where particulate and spectroscopic analytical tools can be deployed to help us better understand these facets of vaccine development. Now, as far as the regulatory landscape, I've always thought of vaccines as kind of occupying a very interesting place in the pharmaceutical portfolio. And that is that the drug substance itself is really a biotherapeutic. So it's typically a naturally derived nucleic acid or some assembly of proteins and nucleic acid that makes up a virus. But the actual delivered product, so the drug product itself, is more akin to a small molecule nanosuspension. And so to me, this actually is a really interesting place to work because it kind of puts together both pieces of my recent background in working in both the large and small molecule arenas. And there are some very pointed documents covering really the drug substance part of the equation. But what about drug product characteristics, especially as kind of a critical quality attribute? And in my reading and preparation for this, I did come across a draft paper from the WHO Expert Committee on Standardization that actually recommends specifically for these LNP delivered messenger RNA vaccines that particle size distribution is measured by one of a number of techniques, including light scattering, NTA, electron microscopy, or SEC. And I took this to be a really nice point to be made that the product quality at the end is going to also be determined by that delivery vehicle itself and whether or not it's stable or if there are any other issues with it. So looking at how vaccines are delivered, again, the audience probably knows quite a bit of this better than I do. But we have a number of different types of delivery. So the more traditional the viral delivery and or adjuvanted vaccines, and then some of the newer vaccine delivery methods, which include LNPs, viral vectors, and virus-like particles. And again, what these all present on some level is really particles. If you look at a virus, it tends to be a bit larger than, for example, a monoclonal antibody. It's a relatively complicated, very large molecular assembly. And so all of these things have some application that particle sizing or particle investigations can help with. Now, what have people been using? Very traditionally, cryo-TEM, not something I have a lot of experience with. DLS, something I have way too much experience with. NTA, so nanoparticle tracking analysis, and then laser diffraction. And what I've shown here is kind of the overlap between those techniques and the size ranges of interest for some of the delivery vehicles that are shown above. Now, I'll be talking a bit more about nanoparticle tracking today. And what you see here is kind of a really saturated image of a bunch of particles floating around in suspension that are scattering a number of different incident laser beams. And what we can do is watch these particles move through suspension by tracking the light that's scattered off of them. And that's really what the experiment is. It's very similar to DLS, except in this case, it's a single particle experiment. Now, what that allows us to derive is the diffusion coefficient, and combined with the temperature that we've done the experiment at and the viscosity, we can directly calculate the hydrodynamic radius or hydrodynamic diameter of that particle. And all that really means is we're assuming it's a sphere, and then there's probably some solvent stuck to it. But it's a very good measure of that nanoscale particle size in these suspensions. Strengths are polydispersed or aggregated samples. It's single particle, so it's highly sensitive on the low end of the size range. It's less sensitive to oversized materials. And we have both national and international standards that govern the deployment of this into labs. So it's really ready to go. Now, what Julie and I worked on for this is more or less just a model system. So instead of using, polystyrene or something like that, which often happens in the particle sizing world, we went out and found a vaccine that we could obtain that was relatively modest in cost. And this is the Canine Spectra 6, which is designed to vaccinate our best friends against a number of different viral diseases. This is a lyophilized sample that includes the diluent with it. And what we did with this, and one quick point there too, there is an adjuvant that's included in this, but we were unable to find any information about that. So we don't know exactly what the adjuvant is, which is the one problem with working over the counter. We did require dilution of the sample in PBS, which is not optimal. We prefer to dilute these obviously in their DP buffer, but that was not available. The recipe for that wasn't available to us. And then we measured this on a 3-laser NTA system. Now, a couple of things about the 3-laser system. Why do we need more than one laser? Well, if you look into the Rayleigh equation, the intensity is the scattered intensity off a particle is proportional to 1 over the wavelength to the 4th power. So as we go lower in wavelength, we get better scattering. So at 635, we can measure larger particles and probably down to the 100 to 200 nanometer range. 520 fills in some of that as well. But with the 445, that allows us to get down to a practical limit with a system like this of about 50 nanometers. And I raise that because I know I've seen specifications for other systems out there that'll say they can go down to 10 or 20 nanometers. And I think that we have to be a little bit careful there because some of that can be specsmanship. Practically speaking, in my lab, the people that I was working with, we had a single red laser system, and we spec'd that down to about 100 to 150 nanometers for biotherapeutics in general. So a little bit conservative on that, but I think that allows us to do better science and to understand things very well. The sample size is 350 microliters, stirred in a cuvette, so nice and easy to clean rather than a flow system. And then it's a fluorescence capable system as well, though we did not deploy fluorescence for these experiments. So what did we see? Well, the first thing we did was dilute in PBS and take a DLS measurement. We got a single mode out of that was centered around 200 nanometers. Now, from my eye, which I've had, again, way too much experience with DLS, this is a very broad mode, and it's also asymmetric, which suggests to me that this is not a monomodal distribution. Now, the resolving power of DLS is somewhere within a factor of 3 or a factor of 5 between two modes to fully resolve them. And one of the things that also bears here is that the intensity-based this is an intensity-based distribution. So there's an R to the 6th intensity dependence. That means that the largest material is going to be far, far, far overexpressed in the experiment. And I think that's what's happening here is we probably have some larger material in here, probably from the reconstitution or the dilution, that's kind of contaminating that result. If we look at an NTA result for this, we see a completely different story. We see a single mode that's roughly centered around 100 nanometers. And this is likely because that the NTA technique, especially with the blue laser available, is capable of measuring the smallest particles that are present down to about 50 nanometers. And that's what we're really seeing. So that's the dominant population in our suspension. If we look at those results over time, so this might be something more along the lines of a stability experiment, or it could even be a formulation development experiment. What we see is that over 40 minutes, we lose 50% of our particle concentration. So literally this last column here in the table shows a monotonic decrease in particle concentration across that 40-minute time frame. Now this isn't something you'd be able to extract from the other types of experiments. And so really access to this concentration information is critical. What I'm showing here in the upper left or the upper right hand, excuse me, panels here, inset panels, is just a smooth version of the actual NTA raw data. And this is more of just a guide, a visual guide for the eye. And really what this is telling us, of course, is that dilution in PBS is destabilizing the formulation. So this highly engineered formulation, we dump in something that really is just kind of water and salt that may not serve the purpose of keeping that suspension stable. So what's the next thing we do? Well, naturally, we try to throw in a little bit of surfactant. PS20 is one that's pretty commonly used in biotherapeutics. And so we added in a 0.02% PBS spiked with this PS20. And what we see is still seeing a decrease in concentration after 40 minutes. But what we're not seeing is that 50% decrease. It's really considerably lower. And also we note that the D90s dropped to about 132 nanometers. So there's something else going on here. And I think in the highest concentration surfactant data, we can kind of start to see what that is. So here we added in 0.2% PS20, and we see that the concentration really hasn't changed at all over 40 minutes. So it looks like we've been able to stabilize this. The D90 is a little bit lower than it was initially for the dilution. And so if we look at the inset here, we can see that this band, that this single population mode is shifting to smaller size, and there's a concomitant increase in concentration of those particles. So really what's happening is its band is shifting and it's narrowing at the same time. So we don't see a drop in concentration, but we're seeing a change in the particle size. Now, this could be due to hidden aggregation. So there could be an unresolved aggregate mode that's sitting under this primary adjuvant mode that we're looking at, or it could be the fact that we're actually disrupting the larger size adjuvant. So you could imagine if this was like an LNP delivery, for example, we might be rupturing some of those. It's not clear. And unfortunately, we ran out of time to do much more work on this. What we did do, however, was some stress work. So we looked at three kind of common stress conditions where we looked at a shaker plate. So we just put a vial of this material with the PS20 on a shaker plate and shook it for 48 hours. It's a pretty easy way to aggregate most therapeutics. 50 degrees C, so accelerated stability was a second condition. And then we did have it in direct sunlight for 2 days. So that's probably more like 24 hours or so of direct UV irradiation. And what we can see is compared to the PBS plus 0.2% polysorbate, that the sample tolerates shaking very well, which is not unexpected. Polysorbate is typically used to kind of stabilize the surface of these biomolecules in suspension, and that helps to protect them against any of this kind of surface charge density that you see when you shake or you have air bubbles entrained into the suspension. The sample also tolerates elevated temperature relatively well, though we still are seeing that shift to smaller size and the narrowing of the population. So that's still not quite explained. And then what we do note is that this does not tolerate direct sunlight at all. So this sample is not stable under UV irradiation, and that's something that would be typically done during a stability panel for the product. So we see a huge drop in concentration. We see some major changes to the band shape and then the particle size as well. Now, moving away from NTA, because laser diffraction was one of the first things I actually worked on outside of the spectroscopic community, is looking at how laser diffraction, so a different type of light scattering method could be deployed here. The first time I saw this working in the vaccine field was about 12 years ago, where I went to for a client visit where they were measuring an hydrogel or aluminum hydroxide adjuvant that they had included in one of their vaccines. And they were using this as a routine analysis tool when they would receive new lots of material and then after manufacture. So that blue curve that you see there is actually an hydrogel sample that Julie and I were able to obtain and measure. So you can see that sits in a really good position for kind of laser diffraction analysis. And obviously that's a very cheap sample compared to a lot of the therapeutic agents that are being built. And so it's more amenable to a larger sample volume, which laser diffraction requires. However, we were able to measure the Spectra 6 vaccine on the same system. And what was cool to note there is that we saw a main adjuvant band down around 100 nanometers, but we were also able to see a larger, what is likely an aggregate or a failed reconstitution band up around 10 microns. And this helps to give us confidence that our assigning of that broad DLS peak was really due to some large material that we didn't want there. And so there is definitely a place, I think, for laser diffraction in these types of applications. It's just something that's widely used in small molecule, but because of the volume constraints and then the size capabilities, it can really go down to somewhere under 100 nanometers. It has not been widely deployed in the large molecule arena. So in summary, just a couple quick points. One, from my experience, vaccines are analytically different. They're really based on a biological product, but the deliverable to the client in this case is particulate in nature. And I think this lends itself well to kind of some of these new technologies. I know NTA has really been around for almost 20 years now, which doesn't seem like that long, but it has. But there's still, I couldn't tell you the number of clients that came to me in the contract world wanting DLS type measurements for their products, and they were steered quickly into using NTA in an advisory role for sure, to help understand what's going on with their product. And then even towards the end, we had a number of clients who were working towards release testing through this NTA technology. So thank you again for your time. I really appreciate it. And again, my contact information is kd@particlesellc.com if you'd like to have a conversation. I'm going to turn the floor over to Linda, who's going to tell you about A-TEEM spectroscopy specific for viruses. And I think this is pretty exciting stuff. So thanks.
Thank you, Kevin, and also thank you very much for for the kind introduction. It's obviously a real pleasure to be working with you again professionally, as well as with Julie. So it's certainly been a lot of fun. All right. So as Kevin noted, vaccines are challenging samples to analyze. Well-established methods can be slow or really not necessarily answering the right questions. And why is this important? Nimble and Bioforum, in their vaccines roadmap from 2018, pre-COVID, pointed out that up to 70% of vaccine manufacturing time is spent in the QC process, and that product release times are typically weeks to months, which could potentially be shortened to 1 to 2 days with appropriate multivariate sensors, multivariate analysis, and predictive modeling. So the need for rapid vaccine release has certainly never really been more pressing than now. But in addition to the release testing, there's also the development, the formulation development that goes on that is also important and also needs tools to speed the process up. So what are the analytical options? They talked about appropriate multivariate sensors. What are our options that might help us in vaccine analysis? So taking a typical vaccine formulation, this is Shingrix, and looking at what goes into a vaccine product. And then we'll see how the kind of standard vibrational spectroscopic tools will fare in looking at this sample. This is a little bit of a hypothetical, but I also have personal experience in using these vibrational tools for vaccine analysis, and I found it very frustrating. And why is that? Well, the first thing we notice is that let's see if I can get a laser pointer going here, is the vaccine concentration, 0.1 mg per mil. That's quite low for vibrational spectroscopy. And then to make matters worse, we have sucrose here at about 100 times. And we know for certain that for ramen, that the sucrose is really just going to overwhelm the protein signature. And oftentimes, for vaccine characterization, the protein signature is what is important. And then we go down to the water-based diluent, and that will be a challenge for FTIR and near infrared. So right now, with our standard tools, we're finding some challenges. So what about fluorescence? Fluorescence is sensitive to proteins, and it is simultaneously insensitive to pure water and sugars. So could that work? So going to fluorescence spectroscopy, if we look at a simple excitation profile or emission profile, just a single spectra, we find that we have somewhat limited information content. Don't get a lot of characterization there. So what do? I'm always one for adding more dimensions. So let's add for every excitation wavelength, we can add a full emission profile, and then we can scan these excitations. And this turns into excitation emission matrix, or an EAM. Standard EAMs are collected by tuning to excitation, and then with a single point detector detecting the points along this axis. And then stepping onto the next excitation wavelength, building up the emission profile. So this can be very slow for collecting a full EAM profile of a sample. So that's kind of the first potential issue we see with this. The other, fluorescence is known for self-absorbance to interfere with the sample profiles, and this is called the inner filter effect. And it can shift the EAM features in a concentration-dependent way. So if you have a fingerprint that is concentration dependent, as coming from ramen and FTIR, I would argue that that may not really be a fingerprint. But it is possible to correct for the IFE using an absorbance spectrum of the sample. That said, having to capture two different types of data, the EAM and separately an absorbance spectrum, is not only time consuming, but also the fact that you're using two different instruments to collect the data introduces error that results in an imperfect correction. So now I'm introducing here A-TEEM, which is a patented approach that solves the limitations of the EAMs that I introduced in the previous slide. The first point is speed. So A-TEEM uses a CCD detector instead of a single point detector. So the entire emission profile is collected at once rather than through scanning over a single point detector. So A-TEEM collects an entire excitation emission matrix in about 30 to 60 seconds, obviously depending on instrumental parameters. So speed is no longer an issue. The second issue that we need to tackle is the IFE correction. So A-TEEM combines the UV vis to get your absorbance spectrum and the EAMs in a single instrument. So the same optics, the same source, the same sample at the same time. So you don't have to worry about the potentially blurring impacts of acquiring these two pieces of information on different systems. So with this kind of inventive tweak, we have two spectrometers really in one box, and suddenly the difficulty in correcting for concentration effects is resolved. And now you have actual fingerprints that you might think of as from some of the vibrational spectroscopic techniques. So the final area of concern is whether this is too much information. Or is it too much information? And certainly, I would argue that in many cases, a single univariate approach to data analysis may get you some answer. It's probably not going to get you all the answer. So careful use of a multivariate analysis allows us to pull the information that we need out of these data sets. And also, because this is a multimodal approach with UV-Vis and fluorescence EAMs together, we actually get more information because we have different modalities in a single measurement that we can kind of sort through to get better classifications. So for biopharmaceutical analysis specifically, we suggest that the Aqualog is the best system. There is another platform that HORIBA has, it's called the Duetta, but the Aqualog, not only does it have the capability for auto-sampling for unattended multi-sample measurements, but it also has the wavelength range that is more in tune with protein analysis, and it has the capability of doing validation. So the USP specifications for UV-Vis and fluorescence spectroscopy are set up as a kind of an automated process with the A-TEEM, so that you can use NIST traceable standards to make sure that your instrument itself is validatable and understand that the results that you're getting follow the USP criteria. So what we have here is by adding UV-Vis, which I've highlighted in amber here, with these attributes to multidimensional fluorescence, or an E-TEEM with kind of these attributes, we end up getting A-TEEM, which shows really good sensitivity and selectivity, as well as up to, again, depending on your sample, part per trillion LODs, which certainly make it a good tool for vaccine analysis. Now, with that quick introduction to the technique, I'll now kind of talk about some of the relevant applications that show off some of the capabilities. The first one is AAV capsids. These are used as vectors to deliver vaccines as well as gene therapies, but we were curious if A-TEEM could be used to, number one, differentiate serotypes, and also be used to predict the empty-full ratio. So these samples were bought commercially. These are reference standards that are commercially available. So again, Kevin raised the point earlier that when you buy samples commercially, you have maybe a little less control over the types of experiments you can run. But we were able to get two different serotypes, AAV2 and AAV9, in the empty or full configuration. So the standards aren't completely full, and I believe they use the transmission electron microscopy for this, and quantitative PCR to give these results. I'm not exact, we're unable to get more information about that. We had to dilute these samples down about 100x with PBS, but those were the samples that we had access to for moving forward with this analysis. So this is the A-TEEM fingerprints in kind of a matrix here. The top row are the AAV2 capsids, bottom row are AAV9, empty here on the left and full on the right. And even by eye, we talk about multivariate versus univariate characterization. Even by eye, you can readily see the difference between an empty AAV and a full AAV capsid. The shift in these features here is not hard to see. Same with the AAV9. What may be less easy to see is whether we can tell the difference between capsids. Your eye might be able to pick up differences in the patterns here, but understanding whether that is quantifiable is important as well. I've summarized here briefly some of the features and where they appear. And what we see here is that for empty capsids, the excitation emission max for these features is a tyrosine-like profile, whereas full capsids, what we have is a quenching and a shifting and something that looks more like tryptophan. So again, we head into multivariate analysis because we'd really like to understand the mechanism of what we're seeing, what's causing these shifts. You can see shifts in protein signatures that might be caused by, for instance, the hydrophobic or hydrophilic environment, or in this case, what we found with a parafac analysis, which is kind of an extended multivariate PCA analysis. So a fairly simple to understand analysis. We see these tyrosine-like component and a tryptophan-like component. Actually, one of the important differences between parafac and PCA is that parafac is, even though it's conceptually similar to a PCA, the component scores and loadings that result typically present actual true sample components. So that's a little different from PCA, where it's more of a statistical variance analysis than deriving pure components. So this helps us in trying to understand what's going on here. So again, we have a tyrosine-like component and a tryptophan-like component. The EAMs that I placed under here are not the parafac factors. They're actually the pure EAMS profile for the empty and full, but it's just to allow you to kind of see how these components are impacting the EAMS that we're seeing. And really, it appears, again, I mentioned this before, AAV empty looks like tyrosine, AAV full looks like tryptophan. So what is going on there? What we believe is that when the payload, the DNA payload, interacts with the protein, it quenches the tyrosine component. And this is important because if we were simply doing a quantitative analysis, number one, DNA doesn't fluoresce. So we're really only seeing the impact of the DNA interaction with the capsid itself. So we're not seeing just DNA and protein component in a sample. We're actually seeing the impact of the interaction. So the fact that the DNA is inside of the capsid is the metric that we have. And for determining whether something is empty or full, that is the metric that you want to have. You want to know whether the DNA that you have is inside the capsid itself. And then with our with the samples that we had access to, we took our full samples and we titrated them down with empties. So we have essentially mostly full and then mixed in empties until we got down to mostly empty. We can see here with basically the Parafax score of component 1, which is that tyrosine-like component. We see this quenching as we're adding more payload to the AAV capsid with a quite nice R squared of 0.997 here. So really interesting quantification of the bound or interacting DNA in the AAV capsids. We also wanted to look at the serotype differentiation. So the technique is very sensitive, and the surface proteins between AAV2 and AAV9 are different. So are we able to differentiate between these two? And what we see for this plot, and this is, sorry, an extreme gradient boost multivariate analysis tool. So this is a little bit of a different technique or different multivariate technique that we applied that uses some machine learning approaches. We see the empty versus full for the AAV9. And then we also see AAV2 empty and full. And the class prediction, so essentially where they fall on this y-axis, shows that we're able to differentiate between these 4 samples. And we also added the titration sequence of the full versus empty, and this was done with just the AAV9 sample, and we're able to see good differentiation between all of these samples. So all of the samples were correctly identified and grouped. We were able to resolve not only the concentrations, but also the different serotypes. And with discrimination, this is kind of summarizing that, the discrimination of the loaded AAVs, at least with that titration sequence that we ran, it's precision of better than 3%. So now moving on to a different type of question. We wanted to look at the repeatability and reproducibility and sensitivity and selectiveness of the technique for vaccines. So this is going back to the canine vaccines that Kevin and Julie ran. And here, rather than focusing on the Soloject 6, we are going to do an analysis of all 4 of the commercially available formulations. So as Kevin mentioned, these are over-the-counter canine vaccines. This chart's a little bit confusing because there are lots of different, it's basically 5-component or 6-component vaccine and a 9-component or 10-component vaccine. One of the important things to notice here is that for 9 and 10, really the only difference between these 2 formulations is the presence or absence of a coronavirus vaccine component. This isn't the coronavirus, this is just a coronavirus, but it's interesting to note that the 9 and 10 component formulations are really quite similar with just a single vaccine component difference. So we wanted to see how reproducible and repeatable is the A-TEEM for characterizing these very similar components. We reconstituted the vaccines following the package instructions. They're lyophilized and we added in the diluent. And then we diluted with DI water. Again, as Kevin mentioned, this isn't necessarily ideal. It would be good to have more of the diluent in order to dilute down, but this is what we had access to. So we measured one set of reconstituted samples on 2 separate days for the reproducibility. And then we had a whole separate series of samples that we ran with a different operator on a different instrument and on a different day to test the model validation and reproducibility. Again, always like to look at the data just to see what the features are. SoloJect 5 actually, I didn't point this out in the formulation sheet, is actually quite different. The 6, 9, and 10 seem to have a very similar type of formulation. SoloJect 5 is kind of an outlier in terms of the excipients, not just the vaccine components. So it has a very different profile, which is not surprising. So here we took the data. We took 4 measurements of the different components. These are the 2 different days, so the same sample, but with samples taken on different days. And just did a principal component analysis, looking at the scatter by charting the 3 different components. And what we see here is that the samples tightly cluster. So the 10s, the 9s, the 5s, and the 6s are very tightly clustered and very well separated. So these 3 components summarize about 93% of the variance in the sample. And this is a really nice differentiation between what these very similar formulations. So we got a new batch of vaccines. We call these the U series, different instrument, different operator. We added these to calibration set, pulled these samples out for validation, and predicted against the model that we had created with the previous sample sets. And what we see here with this confusion matrix is 100% correct classification of these unknowns using a model that was developed on a totally different instrument and set of samples. These are 5 repeats of the 4 vaccine products. So this is really, really nice results that we're seeing here, showing the capability of the technique for reproducibility and repeatability. So really just saying that we can differentiate, do that reproducibly and validatable. So it makes this kind of a very nice tool. One of the things I'm not addressing in this talk is its ability to do quant against either kind of known chromatographic samples, so you're getting a validation set using chromatography, or through running standards. Both of those we've had extremely good luck in terms of doing quantitative analysis. Most of that work is oddly enough in the food side of things. So we've done some wine characterization and taken a 48-hour quality panel for wines and been able to run that in less than an hour and get similar results to the quantitative results. So this is very kind of interesting and exciting. And of course, we love working with the wine samples. So that's very nice as well. So really and I have personal experience trying to characterize vaccines with Raman and FTIR, and the ease with which A-TEEM can tackle this problem is really exciting to me. We were able to differentiate between AAV2 and 9 serotypes, also able to do a rapid empty-full ratio determination of the AAV9, looking at very similar multi-component vaccines, able to do robust differentiation and classification of the different vaccine types. A-TEEM has much lower limits of detection, which is extremely helpful specifically for the vaccine samples. It's able to do this characterization in complex matrices because, again, like the sugars and the water are almost essentially invisible to the fluorescence. So this gives us very good discrimination, and although it wasn't really highlighted in this presentation, other than the empty ratio, full ratio, does very nice quantification. So compared to EAMS as well, because I think many people have tried EAMS and maybe not been satisfied with the results, speed of acquisition is very high. We have that very robust IFE correction to get what I would consider a true EAM fingerprint. And it didn't dwell on this a lot, but this 2-in-1 modality, adding UV-Vis information to a set allows us to get a handle on discrimination that fluorescence might not. So even though I mentioned that some of the components, if they're fluorescence, they do not have native fluorescence, and again, this is native fluorescence, we can often distinguish them by their UV-Vis signature. So this gives us essentially 5 different metrics in order to evaluate differences between samples. None of this is done, obviously, in a vacuum. We have a really great team at HORIBA. Most of this work was collected and analyzed by Adam Gilmore, who's the A-TEEM product manager with HORIBA, and also my colleague, Caroline Chatterday, who is retired from HORIBA. But without their work, none of this obviously would be possible. So thank you very much for your time and attention. Thank you, Julie and Kevin, and we send this back to Julie.
Yes, now is the time to move on to Q&A, and I will turn on my camera. Okay, so so there are a number of questions. So I'll just read it. I have virus-like particles around 20 to 40 nanometers. Can NTA measure these particles, and can you elaborate a little bit more on the particle lower limit of 50 nanometers?
So 20 to 40 nanometers. Realistically speaking, if your sample is extremely clean, you may be able to characterize particles in that size range. Again, it's really once you go down below, I think 50 nanometers, excuse me, things become a little bit sketchy if you have any polydispersity to the sample. Even with the blue laser, there's to my experience, there's not going to be a lot you can do if that's really a real world sample. That's my take on it.
Thank you.
Just jumping in there, because obviously I have particle experience too. The index of refraction for these, if you have an aqueous solution and you have a VLP, the index of refraction is just too similar. It's just the image contrast. This is an imaging microscopy experiment. The contrast that you can get between the solution and the particle is just so small. It can be challenging, but I think there are ways to tackle that with fluorescence. If you can tag them, so that although the benefit is having untagged samples, so that may not be a solution.
I think this is a really good question because this is around the same size as AAV. But okay, how did you come about the name A-TEEM? Can you please explain the meaning of the acronym?
I guess it was written, but I didn't actually read it out loud. It's Absorbance, Transmission, Excitation Emission Matrix Spectroscopy. So that's kind of a mouthful, and I find myself like stumbling over it. So maybe that's why I didn't read it out loud. But A-TEEM, because it combines the absorbance and transmission of the UV-Vis with the EANS of the fluorescence spectrophotometer, we call it A-TEEM.
Thank you. This question actually came in during registration. Could you use NTA to analyze for viral prep, which contain both viral vectors, 80 to 120 nanometer diameter, and exosome, broad ranges from 30 to 300 nanometer, major proportions around 50 to 70 nanometers in diameter?
Yeah, so there's good overlap between the samples. That might be a place where, again, fluorescence would be beneficial. We in my former life, we did use a fluorescence cytometer, which is a different technology. But the reason that contrast mechanism was appealing to us was because we could separate samples. So if we could selectively tag something, for example, we could look at a distribution of grayscale images, and then we could look at a distribution of fluorescence only images, and knowing what our fluorophore was targeted towards, we could easily do a separation there. So that would be my suggestion. The size range, it's on the low end, but it seems like the size range would still be appropriate for the experiment.
And I'll jump in with the exosome application. This is a very common application for the view sizer multi-laser NTA. So this is something that you can certainly email us, and we are happy to take a look and have a discussion with you. In addition to it, we also have the fluorescent capability with the NTA. Next question is, I work with nanoparticles called cubosomes. They can present very squared shapes. So the measurement for the hydrodynamic size would be a sphere that represents the motion of my square particles, with the question mark.
Yep. In a lot of these light scattering techniques, you make the assumption before you ever go into the lab that everything is going to be spherical. It doesn't mean that you couldn't maybe extract some more information, but I doubt with the symmetry of a cube, not something I have experience with, that you'd see much difference from really a truly spherical sample.
Awesome. Is there anything else that you would like to add, Kevin or Linda, before we kind of wrap up?
No,
Thank you,
thanks, Julie.
Julie.
This has been a lot of fun.
It's been a lot of fun.
It's a lot of fun for me too. It's always a pleasure working with you guys. Okay. All right. So I think that's it for now. So on behalf of our particle group, thank you so much for attending. Thank you so much, Kevin. Thank you, Linda. And hopefully we'll see you guys at our next webinar in September. So don't miss it. Okay. That's it for now. Thank you so much. Bye,
Take care.
everyone.
Bye.